Welcome 12th class students, Mrs. Nanda biology tutorial. Today the topic is fertilization in flowering plant. Previously we have discussed up to the self-pollination and cross-pollination. To prevent the self-pollination, then the various devices are outbreeding devices. Outbreeding devices. That means dioxy or diclining. Both plants, male and female, are separate or unisexual. So it means they prevent self-pollination. That is a first part. Second device, self-sterile. Certain genes present in pollen grain as well as pistil, so they are incompatible for fertilization, that is S1, S3 genes. So then the condition is called self-sterility and they prevent the self-pollination. Third criteria or third device, that is called to be as dichome. G-A-M-Y. Dichogamy. That means there is a maturity of this male and female or pollen grain or pistil at different time, or stamens at a different time. The condition is called protandry and protogynous. Protandry and protogynous. Protandry means stigma mature earlier than, so there is no self-fertilization. Protogamy means protogamy, that is female part mature earlier. So that condition, dichogamy, prevents self-pollination. Why we want to prevent self-pollination? Because every time they give the same character, same race, and they also give the inbreeding depression, continuous race. So we have to, for this control, we have to undergo this outbreeding devices. In NCERT it's a separate topic and definition can come. Now come to the point, the fertilization occur when they are special pollen pistil interaction. This is very important. Pollen pistil interaction, because stigma is sticky in nature and pollen grains come in contact after pollination. So shedding of the pollen grain, as we have discussed, two cell stage after the pollination, that is like this. So there is a complete shedding of the pollen grain, come in contact. Now the further changes, if they are compatible, that means pistil and pollen are compatible of the same, so then they undergo the phenomena called fertilization. If they are incompatible, so there is no germination of pollen grain on the stigma and there is no formation of pollen tube. But if they are compatible, then they undergo the process called fertilization. So fertilization is fusion of the male gamete with female gamete, and it is also called syngamy. So how it occurs, now this is a diagram showing LH or longitudinal section, how the pollen tube is formed in the stage. This is the stigma, style, this is style, this is ovary. This is called to be pollen grain, the labeling. Again important for your board exam, the pollen grain. So the germination. Now the structure, that how it happens, we have discussed the ovule structure. Now according to NCERT, this diagram is important. Longitudinal section of the flower showing growth of the pollen tube. That is important for you people, for the board. Now LS of the flower. So what is the movement or how they are formed? So pollen tube is formed, that is a male gametophyte, and this pollen tube carries two male gametes, as we have discussed. Two male gametes are carried by this pollen tube. So now this pollen tube, with the fertilization through pollen tube, then it is called siphonogamy. It is called siphonogamy when fertilization through pollen tube. Now, if they are compatible, pollen tube is formed and break the tissue inside the style and by the action of certain chemicals. Now, in the ovary, the tissue which give the path or direction to the pollen tube is called obturator, which give the pathway to the pollen tube to enter into the embryo sac. Now, this is the ovary and this is a single ovule we have discussed, surrounded by integuments. Now, inside this ovule having embryo sac. So, embryo sac means consists of the 8. Discuss the structure. Embryo sac, towards the micropyle, egg, followed by synergid, egg or synergids. In the center, we have discussed pollen tube, pollen nuclei surrounded by a cell. Secondary and 3 are antipodes. This is the inner structure of an embryo sac. Now, what happened when the pollen tube enters? Pollen tube enters. It enters through synergid, and synergid form a special structure called filiform apparatus. Give the direction to the pollen tube. So what happened? They can be entered either mostly through the pore, that is a micropyle side. This is the chalazal side. When they enter through pore, then the condition is called porogamy. But when pollen tube enters after breaking this integument, this is the outer integument, when they enter through this, then it is called mesogamy. If they enter through integuments. If in certain condition, sometime they enter through the upper end, chalazal end. This is the chalazal end. This is the structure of just embryo sac. This is the micropylar end. So when they enter, then this is called chalazogamy. So entry of the pollen tube into the embryo sac by means of porogamy, mesogamy and chalazogamy. So what happened? The filiform apparatus, that means this is the filiform apparatus, give the direction like this. So when they enter like this, they release 2 male gametes inside. Release of 2 male gametes inside. Now, one combined with 1 male gamete combined with egg to form the structure called zygote. To form the structure called zygote, which is diploid. Which is diploid in nature. 2N structure. Secondly, and the second, so it is formed with egg, or it is also called oosphere. So mind it, oosphere before fertilization, oosphere, and oospore after fertilization. So first structure is formed, that is called to be zygote. Second male gamete fuses with 2 polar nuclei which is present in the center. 2 polar nuclei present in center to form the structure called endosperm, which is triploid. It is also called to be triple fusion. So 2 structures are formed simultaneously. Then this phenomena in angiosperm is called double fertilization. It is called double fertilization because 2 structures are formed simultaneously. One is zygote, second is endosperm. The fertilization to form zygote is also called generative fertilization. It is called generative. Endosperm, another structure is formed which is called vegetative fertilization. Together constitute this double fertilization, and this triploid structure endosperm is also called PEN. when the secondary nucleus combine with polar nuclei to form PEN, primary endosperm nucleus, or the cell is called that PEC, primary endosperm cell. So it means this double fertilization is main feature of the angiosperm 2 structure. So endosperm provide nourishment to the developing embryo. We will discuss this endosperm in the next video. So 2 structures are formed and 2 fertilization are formed. This is called to be the fertilization. Both are very important for the board, fertilization, double fertilization. Then comes sometime, this is another subtopic called artificial hybridization. Hybridization is quite common to improve plants variety. Plant varieties quite common in and to improve the plant varieties or crop improvement program. In this crossing over experiments, in this, what is a major approach to improve crop variety? Whether they are resistant to diseases or more yield production. So in this case, desired pollen grain are used artificially. Desired pollen grain or healthy character pollen grain are used. Stigma is again protected from contamination. So what happened? 2 main techniques are used. One is called emasculation. That means emasculation is the phenomena. It occurs in bisexual. So removal of the anther before their descent from their flower bud. This phenomena or this technique is called emasculation. Definition 1 mark for the board. Second, bagging. What they do? They cover after descent. They cover this flower with a polythene or butter paper to protect. There is no self-fertilization. So it means always with the cross or desired variety or pollen grains are used. So this technique can improve the crop variety by this technique. So that is called to be artificial hybridization. That means done by the human being or to improve the crop production. That means that the better variety, more yielding varieties are formed of the plants to give the more yield production or high yield production. I think it's clear. Thank you.